Jason Cantley, excuse me.
Okay, well, thank you, Ingrid, for an introduction, and you are also a co-author on this paper, and I should also recognize that 2 undergraduates were part of this research, and without them, it really wasn't going to be happening. So, I want to move away from mosses for a moment and show you a flower. It's a really beautiful flower, and it's sort of the epitome of millions of years of evolution. We see that we have different parts here in this flower, a yellow area which has all the male parts, and a green and white structure here that allows for fertilization and pollination, and we call this female. This flower actually represents a genus of plants, Solanum, which the genus is worldwide. It's about 1,500 species, and it is quite remarkable. And I also want us to start thinking about the differences of male and female parts, because this is really interesting because there's different breeding systems and different ways for this lineage of plants to share its genetic information with other individuals. The most common breeding system in Solanum is probably hermaphroditism. And this is sort of the common standard. You can bring pollen in and cause an outcrossing event, and you can also do a selfing event at the same time. Okay, really key here is you can self and you can outcross. We also have this slightly less common situation in Solanum, and this is an andromonoecious breeding system. And in this breeding system, we have a solitary inflorescence which has a basal hermaphroditic flower having both male and female parts. And then above that, we have many male flowers that generate, that grow above it. And what's really interesting is that you can also have a selfing event, but you can also take this pollen into outcrossing. So it's very similar in a lot of respects to the hermaphroditic breeding system. The least common breeding system in Solanum is a dioecious breeding system, where these male parts and female parts actually occur on different plants. And this lineage is developed a few times in different places in the world. But what's really cool about it is the pollination direction can only happen in an outcrossing event from a male plant to a female plant. And if you're looking really close, you might see this little area here around a lot of male stuff and say, wait, that's actually a hermaphrodite. But as it turns out, there's some really cryptic morphological characters that make this flower functionally female, even though it presents itself as a hermaphrodite. And here what we see is that pollen grains in that functionally female flower actually never develop the final pores that allow the pollen sperm cells to germinate and cause effective pollination. So we have these different ways of spreading genes to the next generation of flowers in these different breeding systems. And this has led us and some other authors to hypothesize that there would be differential expectation of the genetic diversity in these populations. In these breeding systems. And if you look at that, you could say that an andromonoecious and a hermaphroditic breeding system would share a similar level of genetic diversity because they are able to self and also outcross. But because dioecious breeding system cannot self with itself, then we see that we would expect maybe some higher genetic diversity. And so this is really the fundamental underpinnings of our study that we are looking at. And what's really cool about this is we have a lot of new information that is showing that in the same place in this Australian spiny Solanum lineage, we have 2 derivations of a dioecious breeding system. So this is the phylotune of a lot of different work using low copy nuclear stuff as well as a concordant study on B and RADSEq. And we see that the breeding systems represented by A, H, and D show a transition to a dioecious breeding system, which that's what the D represents here twice. And these breeding systems also occur in the same exact physical place in the northern portion of Australia. Now that's really cool because these plants fundamentally only differ in their breeding system. They occur on the same monolithic outcrops. they have the same sort of structure as plants. I mean, there's a little bit of size difference, but fundamentally, their primary difference is that they have these different breeding systems. So today, I wanted to talk about 5 taxa. Three of them are the dioecious breeding system, one hermaphroditic breeding system, Solanum echinatum, and the final one that's andromonoecious Solanum clarkii. And this work was done over many years, starting in 2009, really more than a decade, more or less. And there's lots of people to thank. The work in the field for this study is taking 20 of those populations from those 5 taxa and looking at these population genetic information that we can obtain from that. So once we left Australia and we got back to the lab in Pennsylvania, we did methods and methods and tons of methods. So essentially, I'll distill it down to enough so that you can get a pipeline understanding of what we did. First, we did a DD-RADIC protocol, and we did filtering, and we took 365 individuals, and we did all the awesome wet lab stuff. We had the data come back to us, and we did a PIRAD and a pair demultiplexing and a de novo assembly of the loci. We did descriptive statistics to look at FST and pi and heterozygosity. We did a phylogeny using Bayesian inference and maximum likelihood, which was over thousands of loci and over 1.5 million base pairs. We did admixture tests in structure, which was over 15 different runs to look for hybridization between the taxa, as well as admixture occurring between the populations of a single taxon. And finally, we're actually really interested in looking at demographic history. So what were the bottlenecks, if any, in populations, and what other kinds of information can we learn about the effective breeding sizes of these individuals? So that's a lot of stuff, and I only have a little bit of time to share some of the really cool findings. And on the right here, I have some descriptive statistics that are a little preliminary, so take those with a grain of salt as we're still working on filtering the data to an appropriate state to show them. But what I wanted to show is that the hermaphroditic breeding system and the andromonoecious breeding system that were hypothesized to have a similar level of genetic structure actually do. So they're very similar. And these structure bar plots, which I should first let you know that these 2 plots were done in totally separate runs, so the colors do not compare across bars, but you can see that they have a similar exchange of genetic information if you're used to reading them. And the vertical lines here, actually, on my computer I can see separations for each of the individuals, but the vertical lines actually represent an individual, and the colors represent a haplotype, more or less, is a better way to explain it. Now, what's really cool is if we look at the dioecious breeding system, we see a really different pattern. Okay, our descriptive statistics suggest that there is a much higher genetic diversity for this breeding system and three of the taxon that we tested, and we see that there's a greater amount of genetic admixture between the populations of a single taxon. Now that's amazing, and that is what we would expect, right? These things cannot self, so they are obligate outcrossers. And that signature, I think, is what we're actually really picking up on in these analyses. And then I just love this slide because this is showing the populations and the admixture of these taxa, but kind of comparing across at certain populations that are in the same physical spot. So Merle Rock A has a hermaphroditic taxon, Solanum echinatum, and it has a very homogeneous population structure. And in the same location, Solanum asymmetrophyllum, that's dioecious, has a lot of admixtures with very nearby populations, and it has a higher genetic diversity, as inferred from our descriptive statistics. We can also look at this in actual physical space. Now, the black lines here actually are a sandstone monolith that rises maybe a few meters in the air in Kakadu National Park in the Northern Territory of Australia. And we can see that we have a different structure of the population for the red dotted Solanum asymmetrophyllum that's dioecious in comparison to the green Solanum echinatum, which is hermaphroditic. And finally, I really was interested in this. Okay, this is cool, but can we actually look at things that were happening in the past and correlate a similar response to environmental conditions in the past for these different taxa? So we use this program called GFOX that looks at all the loci that were generated from our RADSEQ, Agidi RADSEQ protocols, and we analyze them to map a demographic history for these individuals. Now the way to read this is we have present time at the lower portions of these diagrams, and it increases in relative time. We've not scaled this to actual years, to deep time in the past. And on the x-axis here, we have the effective population breeding sizes for these individuals. And we can see that when you have a constriction, we have an inferred bottleneck. Now that could be from range restriction or maybe dispersal to a new population. So lots of different things to think about with these models. But what's really cool is when we scale this in relative time, we see that there's a hugely different history for these individual taxa. And you can first see that Solanum clarkii, which is our andromonaceous taxon, has way deeper divergences between its populations than the other ones that are really small down here. We also were able to look at post-divergence gene migration. So this is migration occurring between the populations after they have already diverged. And we see that there's really a negligible amount of that occurring for all of these taxa. And I should also, let's zoom in, and these are, I want to mostly show that how recent this is. They're really new taxa. We're working in a time frame of probably less than 1 million years from some external analyses. But now we have this really cool comparison of Solanum asymmetric phylum that's a dioecious taxon, and Solanum echinatum, which is hermaphroditic. And they occur in the same plot, in the same places, in at least 3 of the populations. And we're seeing a really cool pattern that we can look at and think about in a way, because we see that we have almost the same time for these bottlenecks when they occur, but there is a differential response to rebounding in the Solanum asymmetric phylum, which we now have FST values suggesting there's a greater genetic diversity in these populations. It's really fascinating and tantalizing results, and I'm really looking forward to doing some more work with this and analyzing this a little bit more specifically. Lastly, I really want to talk about isolation. So the farther these populations are, it seems like there's more unique haplotypes occurring for these individuals and less outcrossing. So one of our next steps is to look at isolation by distance using mantle tests and EcoDis package of R to really get a better understanding of what's going on at that level. So if there's anything you should take away from this talk, it's the idea that a dioecious breeding system, at least in the lineage of Solanum and the northern territory of Australia, does pay to be dioecious. Okay, we have populations that occur in the same place, and the dioecious taxa are showing better, higher levels of genetic diversity, especially when our population sizes are large, and both sexes have to be there. But also, some data I didn't present to you was a really small taxon that's dioecious, and it's small and isolated and produces clones. Then we lose this pattern or the benefits of dioeciousness. So with that, I'd just like to thank the Martine Lab. They're amazing, tons of undergrads and a master's student, and we could not do this work without the help of all these undergraduates. It's really quite an amazing experience and lab to work at. I also have to thank a lot of my funding sources, and so thank you.